Gene editing. What's possible now? What's banned? What's next? CRISPR gene editing has achieved a significant milestone. It is no longer merely a lab capability searching for a patient. Today, edited cells are a regulated and marketed medical product in multiple countries, providing treatment to real people. The hype is still ahead of the reality, but the reality is now big enough to force policy, pricing, and ethics decisions that can't be postponed. The central tension is simple. Gene editing can be permanent, but biology is messy. That makes the upside enormous, and the failure modes unusually sharp. The overlooked hinge is that the limiter is less often the editor itself than the delivery system, and the evidence standard regulators will accept when outcomes take years to fully surface. The story turns on whether gene editing becomes a scalable platform or stays a boutique craft. Key points. Gene editing is already treating patients through ex vivo approaches, where cells are edited outside the body and reinfused. This is the most mature path because delivery is controlled. In vivo editing. Editing inside the body is advancing fastest in organs that are easier to target, especially the liver, but safety events and immune risks can slow timelines quickly. What's banned is mostly about reproductive germline editing, heritable edits, not about treating disease in an existing person. Many jurisdictions allow embryo research under strict limits while prohibiting implantation. Off-target edits are only one risk. On-target unintended outcomes, immune reactions, and delivery vector toxicity can matter just as much in real-world trials. The next leap is not a single breakthrough tool. It is a bundle of compounding improvements, better delivery, better control over outcomes, and clearer regulatory playbooks. Realistic timelines are split into two tracks: faster for single-organ targets with good delivery and measurable biomarkers, and slower for brain, muscle, and complex common diseases where targeting and follow-up are harder. Background. CRISPR is best understood as a programmable GPS-guided molecular tool. A guide sequence brings the editor to a chosen DNA address, and an enzyme does the work. The original CRISPR-Cas9 approach usually cuts DNA, then relies on the cell's repair machinery. That can disable a gene, or it can be used to rewrite sequences, but repair is not perfectly predictable. Gene editing now includes more than CRISPR scissors. Base editing changes a single DNA letter without making a full double-strand cut. Prime editing is more like find and replace, designed to write more precise edits. These newer tools strive to enhance the scope of repair while mitigating the potential harm that cutting may cause. The crucial practical split is somatic versus germline. Somatic editing changes cells in a living person and is not inherited by their children. Germline editing changes eggs, sperm, or embryos in a way that can be passed on. Most current clinical momentum is in somatic editing because the ethical and regulatory barriers are lower and because benefit can be judged in the treated person. Delivery is the other foundational concept. Editing tools do not matter if they cannot reach enough of the right cells. Today, the main ways to deliver treatments are lipid nanoparticles, which are often used to target the liver, viral vectors like AAV, and methods that involve physical or lab-controlled delivery in ex vivo. Analysis. What's possible now in humans? The most proven model is ex vivo editing of blood-forming stem cells. Cells are taken from a patient, edited under controlled conditions, quality-checked, and then reinfused after conditioning therapy. This model is logistically heavy, but it solves the hardest part. You can confirm editing happened before the cells go back into the body. This strategy is why the first widely recognized regulatory firsts have been in blood disorders. The clinical logic is clean. A corrected blood stem cell can repopulate the blood system, and clinical endpoints are measurable. In vivo editing is no longer theoretical. The liver serves as the current focal point due to the relative ease with which nanoparticles and some vectors can reach it, and the ability of blood biomarkers to confirm the effective alteration of a targeted gene. But in vivo work is where safety events reshape the pace. When the editing system is delivered system-wide, you inherit every uncertainty about distribution, immune activation, and unintended edits in non-target cells. What's still hard? Delivery, precision, and control. Most public discussion still overweights off-target edits, but the real-world risk picture is broader. One category is on-target unpredictability. Even when the editor hits the right site, cells can repair cuts in different ways, producing a mixture of outcomes. That can be acceptable in some diseases and unacceptable in others, depending on how sensitive the biology is to exact sequence changes. Another category is delivery toxicity and immune responses. Viral vectors can trigger immune reactions and have limits on how much genetic payload they can carry. Lipid nanoparticles have their own tolerability ceilings, and doses not free. Higher exposure can raise risk even if it raises editing efficiency. Then there is tissue access. Blood and liver are reachable. The brain, heart, skeletal muscle, and many solid organs are more challenging to access. Common diseases often involve difficult-to-target tissues, multiple genes, or complex causal chains, making a single edit ineffective. The distinction between somatic and germline editing represents both an ethical and a technical boundary. Somatic editing is moving because its moral claim is familiar. Treat an illness in a consenting patient. Germline editing is different because it creates an irreversible decision for a future person who cannot consent, and because mistakes would echo into future generations. But the ethical boundaries are not the only ones. Germline editing also demands a higher proof standard. You would need extreme confidence not just in the edit, but in how that edit behaves across a lifetime and across diverse genetic backgrounds. Earning that kind of proof takes a significant amount of time. In practice, many places differentiate between embryo research and embryo implantation. Research that edits embryos for early developmental study can be permitted under strict limits, while implantation for reproduction remains prohibited or blocked. That means banned. It often means banned to make a baby, not banned to study in a lab. What's banned, what's regulated, and what's in the grey zone? Across many jurisdictions, reproductive germline editing is treated as out of bounds. The mechanisms differ. Some countries use explicit criminal law, some use licensing and regulatory enforcement, and some rely on funding restrictions and the inability to legally run a clinical trial pathway. The United States is a useful example of enforcement by process. Even without a single comprehensive federal statute, policy riders and regulatory barriers make it effectively impossible to seek lawful approval for heritable genome editing intended for pregnancy. Meanwhile, somatic gene therapies are regulated through established drug and biologics pathways, which is why clinical development continues there. The grey zone is expanding around embryo models and early development research tools that are not quite embryos, but can mimic parts of embryonic development. Regulators are being forced to clarify what counts, what oversight applies, and what time limits should govern research. Those clarifications matter because they shape what can be learned about early human development and what forms of future reproductive intervention might become technically plausible. even if politically unacceptable. What most coverage misses. The hinge is that gene editing's future is being decided less by can we edit DNA, and more by can we deliver edits reliably, and can regulators certify a platform without relitigating the entire risk story every time. The mechanism is compounding. Each incremental gain in delivery, targeting, manufacturing, and monitoring does not just add capability, it multiplies it. Better delivery improves efficacy, reduces dose, lowers toxicity risk, and simplifies trials. Clearer evidence standards reduce uncertainty for investors and health systems. Together, those changes shift which diseases are economically viable to pursue, which hospitals can administer treatments, and how quickly regulators can evaluate follow-on therapies built on similar components. 2 signposts will reveal whether the platform era is arriving. First, watch for regulators formalizing more consistent guidance for in vivo editing safety monitoring and long-term follow-up, creating a clearer playbook for developers. Second, watch for more approvals or advanced stage trials that rely heavily on validated biomarkers as primary evidence of benefit, because that shortens the time between proof and access. What happens next? In the near term, the most affected groups are patients with severe single-gene diseases, where the biology is well understood and where edited cells can plausibly provide durable benefit. The short-term story is access and throughput. How many centers can deliver complex ex vivo workflows? How quickly manufacturing can scale? And whether payers will support high upfront costs. Over the longer term, the biggest shift will come from in vivo delivery becoming safer and more predictable, because that is what turns gene editing from a few thousand patients into potentially millions. That path will be uneven. Safety events can halt programs, and every new delivery system has to earn trust. The main consequence is a widening gap between what is technically imaginable and what is practically deployable, because medicine is constrained by logistics, regulation, and cost, not just science. Real-world impact. A hospital system builds a cell therapy lane with dedicated staff, isolation rooms, and a supply chain for patient cells, because the bottleneck is no longer a drug vial. It is a coordinated workflow with zero tolerance for error. A family with a child eligible for an approved ex vivo therapy faces a different kind of dilemma. The promise of durable benefit versus the reality of conditioning chemotherapy, time in specialist care, and uncertainty about long-term outcomes. A biotech team designing an in vivo edit spends more time on delivery chemistry, immune profiling, and toxicity monitoring than on the editor itself, because a perfect edit that cannot be safely delivered is commercially dead. A regulator faces pressure to move faster for rare diseases with no alternatives, while also knowing that a single high-profile adverse event can damage public trust and slow the entire field. The new contract between biology and society. Gene editing is steadily rewriting what medicine can promise, but it is also rewriting what society must decide. The nearer the technology gets to reproduction, the more it stops being a clinical question and becomes a governance question. Who sets limits? How are those limits enforced? And what level of uncertainty is acceptable? The next phase will not be defined by a single cure. It will be defined by whether gene editing becomes routine enough to be boring, backed by standardized delivery, repeatable outcomes, and enforceable rules. Watch the delivery breakthroughs, the safety holds, and the regulatory playbooks. That is where this moment becomes history. For more insight and less noise, subscribe and explore the full range at taylortailored.co.uk.